Hey, brother, hear me now. Brother dog, know me, understand.
Welcome to the Sargassum Podcast. I'm Robbie Thigpen.
I'm Francisca Elmer.
And I am Mar Fernandez. And we are your hosts for today. And we are going to share with you the latest ideas and concepts about sargassum and sargassum beaching events which have become an international challenge.
Good morning, everyone. How you doing today? I hope you're doing fine. We appreciate you coming and hanging out with us today. We got a couple of more interesting guests from a couple of different places today. And, of course, we got Cleo and Francisca with us. One of the young ladies is with us, Lindsay. She's currently in D.C., which is not a bad city to live in. Additionally, we have Amy Suida there, and she's usually in Florida, but currently she is inside the Blue Line in upstate New York on holiday, and we really appreciate her time, especially today, since she's taking time away from the family. And anyway, Francisca, Cleo, if anything interesting happened to you folks this week?
Yeah, pretty interesting. I went to Isla Mujeres to swim with whale sharks, and I got to see a lot of sargassum on the way to the whale sharks. And Isla Mujeres is one of the few places here in Mexico that doesn't have sargassum on the beach, because the beach is facing a different direction. And coming back here in Playa, the beach is pretty clear now. Like, a lot of the beaches are cleared. The resort, Robbie, you were so outrageous about that they don't clear the beach. The beach is clear. However, the water is still brown because there's so much sargassum on the sediment and still leaching all that brown stuff into the water. So still no swimming here.
That sounds really interesting, Francisca. I wish I could be there too, swimming the shark and the sargassum.
Yeah, that was pretty
in
cool.
The sea, which is actually really wonderful, I guess. For me, nothing so new, so I will let you, Robbie, give us some news about your week.
Oh, my week is, I'm the name of mediocrity, so I haven't really accomplished too much either. I got home from visiting Quintana Roo, and all that was really nice. And I had a really nice conversation last week with this nice professor from Eckerd College about our plastics aspect of the project, and that would be one of our guests today. And with that, I'm going to hand it over to Francisca, and she can introduce our guest, and we'll get started with the interview.
Thank you, Robbie. So our guests today are Lindsay Martin and Amy Suida. Lindsay is a science assistant at the National Science Foundation who is passionate about combining research with education. She has quite literally sailed around the world and is working on making research more equitable and accessible. Amy Suida is an associate professor of marine science at Eckerd College in St. Petersburg in Florida and a former professor and chief scientist at Sea Education Association in Woods Hole, Massachusetts. She is a marine ecologist and biological oceanographer whose research interests span marine plankton ecology from organismal to population and community levels. Lindsay and Amy just published a paper on the motile animals found on different types of pelagic sargassum. Welcome to the podcast, Lindsay and Amy.
For having us. Excited to be here and to talk about this paper, which has been multiple years of effort. So.
Yes, thank you for having us.
So, hello, Lindsay and Amy. Nice to meet you.
Hi, Cleo.
So we will start with the first question. Our first question: what is sargassum to you?
Can I start, Lindsay? Is that all right? So sargassum to me, I have a long history of observing sargassum from sailing with Sea Education Association starting in the mid-90s up until about 2016 or so when I moved to Eckerd College in Florida. but sailing through the Sargasso Sea once or twice a year, sailing in the Caribbean and seeing sargassum from the boat. And so to me, it's this amazing ecosystem that is so different from the water around it. And so it provides this just unique environment, holds these unique organisms that are so different from the waters around it. And so it's really exciting to see it out there in the open ocean, this yellow expanse on those deep blue waters.
And for me, sargassum is a bunch of different things. It was my trip with SCA, with Amy, was my first marine biology experience, where I did my college degree, was a liberal arts institution, so it didn't have a very specific focus on marine biology. So SCA was my first time really doing any research on the ocean. And we were studying sargassum. So that was my first time. It's my intro. It is from a artist or photographer's standpoint, which I like to think myself as, absolutely stunning. As Amy said, it's completely opposite colors. It's the deepest blue purple water you can ever imagine, combined with gold. And it's at the surface, so there's no color correction to worry about. It's absolutely perfect as a photography subject. And yeah, it's just been part of my life now for almost 10 years in some respects. And so I'm, very grateful. Not only is it an amazing ecological environment, it's been great for my own life as well.
Well, Amy, it sounded like you have been watching some of my videos, because you sound exactly like how I describe this amazing ecosystem. When I find a clump just washed up on the beach, and I really appreciate that. And Lindsay, you too. I love that deep blue. There's nothing quite like it, and it's so opaque and transparent at the same time. And I hadn't seen the Sargasso Sea before. I've seen some huge mats off the coast of South Carolina, so I empathize with a lot with your words. However, let's get back to the big part of the interview instead of me just blowing smoke. Your research was looking at motile epifauna communities, and how would you describe this to just a regular old person?
Sure. So to break down those three words, motile is a fancy way of referring to an animal that is able to move around under its own power. So you have something like a sea anemone, which is attached to a substrate, the ocean floor, a rock, what have you. A crab is something that is motile. It can crawl around. Epifauna, so fauna meaning an animal. Epi meaning something that is, it needs to be attached or crawling around on something else. And community refers to the collections of all living things in an area. So what's really interesting about the sargassum community is that these are all animals that you would normally see, say, in somewhere like a tide pool or on the seafloor. These are crabs and shrimp and snails and animals that are very closely related to a seahorse, which uses its tail to hold on to things. So these are organisms that have to be attached to something, have to be near something to live. So it's really weird to find them in the middle of the ocean, at the surface of the ocean, and that's what makes this particular community really unique. Is you've got these animals that aren't supposed to be there but are. So these animals, the ones that we were studying, are the ones that are crawling around on sargassum as it floats through the middle of the ocean. And there are also attached flora, so plants and fauna, other animals. We didn't study those as part of this particular paper, but those are things, there are tiny anemones. But all those little encrusting organisms that you could find on a buoy that washes up on shore. You can find very small versions of that on Sargassum. So we didn't study those. We studied what we call the clinging fauna. So things that have to crawl around on the Sargassum. So those are our snails and shrimp, which were the most common species, crabs, sea slugs, otherwise known as nudibranchs, pipefish, which are related to seahorses, and frogfish, which kind of look like an anglerfish. But then there's also this larger ecosystem that is reliant on the Sargassum in some way. Maybe doesn't stay there permanently, but these are things like transitory fish. So, large fish that are just swimming around in the middle of the ocean are using Sargassum as hunting grounds. But then you also have things like sea turtles or birds or even eel larvae that use patches of floating Sargassum as a refuge, a place to float, a place to hide, because there's not much, there's not much of an opportunity to be able to do that in the middle of the ocean.
One of the species that I like to use as an example to show connectivity between these vastly, separated environments is the Atlantic eel. They come up in these rivers and swamps right here where I am, and then they go out there to spawn. And so they have a very fascinating life history, and so so I like to talk about them every chance I get, just to show this symbiotic relationship between terrestrial ecosystems and the sea.
Yeah, and especially for commercially important species, like any fisherman in the Gulf of Mexico knows to go towards a Sargassum mat if you want to grab a fish. There are just schools of mahi underneath those mats in the Gulf of Mexico, and you can catch 30 fish in the space of an hour if you need to. So they're incredibly diverse and really good at concentrating all of that life that you find out in the middle of the ocean, which is so dispersed because there's just nothing out there. Sargassum concentrates it.
And I think we focused on these clinging fauna because they are that link between the Sargassum community itself and the broader oceanic food web. And so they're making that connection, and they're available as food to those smaller fish and then larger, ecologically and economically important fish.
Wow, that's really cool. I think definitely learned a lot about the different animals that are there and how you put them in different groups. We also have a question from our Patreon, Mark, actually a few questions, but this is the first one he submitted for this interview. So for our listeners, if you ever want to know who we interview in advance and submit questions, go to our Patreon site and become one of our Patreons, and you can also submit questions early on. So Mark, he is from St. Martin, and he loves to look for critters in the Sargassum when it comes to shore or close to shore. So he wants to know, how can one best get the critters out of the Sargassum for analysis, and how do you make sure you do not miss any of them?
So I'll start by sort of describing the way we sampled. We were sampling offshore. So to grab a clump or an individual of Sargassum, we were aiming to do that with using a dip net. So think of like a pool skimmer, and it has a fine mesh or 333 or 335 micron mesh, so a third of a millimeter mesh on that. And we likely are not losing those modal fauna by using that size mesh. They're larger than that size. And we'd aim for collecting a single macroalgal organism, which is what we are terming a clump of Sargassum. Those clumps can then aggregate into larger mats, but we were aiming to collect individual clumps to understand the community on those and on the different types of Sargassum. And so when we collect a clump, we dip over the side of the boat with this dip net as we're moving slowly, collect a single clump, bring it aboard, move it to a bucket of seawater, actually, and then collect all of the organisms that may have come off pretty quickly in the net and make sure we examine the net carefully and pull all of those out. But then we take the sargassum that's in seawater once we've collected it, and we actually submerge it and rinse it in fresh water. We found that that has helped to get any of those really stubborn, clingy organisms off the sargassum. So the snails are often really deep into a sargassum clump toward the center, and they're sticking really well. And so to get them off, it helps to put the organisms in fresh water, to put the sargassum in fresh water to get things to sort of fall off of it so we could capture the whole community. And one thing that we did find as well, there are a number of, most sargassum modal fauna are cryptic in their coloring and they look a lot like sargassum. So it helps to have them fall off the sargassum in order to find them. There are also some clear organisms. So I know the pycnogonids, the sea spiders, they seem to be more common in our Sargasso Sea samples than other locations, and they're pretty rare. But when we did find them, it was more by looking for shadows in the bucket after they had fallen off, because they were difficult to find. So it's a lot of careful looking through that bucket, then taking the sargassum itself and literally looking everywhere along the blades and the stipes to basically make sure we are capturing the whole community to examine.
Wow, this is really interesting. Actually, you already answered one of my questions about the collection methods, so I'm going to take the other one. The question from our patron, Mark. You found differences in diversity and abundance between small and large samples. Would you suggest for future research to also collect both small and large samples, or just one of these kinds?
Sure. So I kind of want to specify what we defined as a sample. So we, when you see sargassum or lots of pictures of sargassum, it's typically these really large mats or windrows. So those nice long rows of sargassum. And those mats or those rows are made up of what Amy said, a clump. An individual clump of sargassum. And you can think of that like, say, a bush that you have growing in your backyard. The bush can be small, it can be large, but it is one plant. And if you have several bushes together, it's a hedgerow. A bush can't move, obviously, but a clump of sargassum is that individual unit that can move around. A mat can be dispersed. If the wind patterns or current patterns change, the mat can break up, and it's broken up into individual clumps. So we sampled those individual clumps and said, okay, this is one sample. We're going to count the types and numbers of animals that we found on that one clump. And because it's a living thing, it grows. And because of, wave action or wind, they break up. So they are different sizes. So that's where the different size of our samples came from. And we found, as you would expect, that the smaller samples had different sized communities than the larger samples, just like a sapling tree is going to support a smaller number of animals living on it than a 200-year-old tree. So one of the ways we analyzed that was looking at how the number of animals and the number of species changed as our sample mass got larger. And we found, as you would expect, they did. So as a clump of sargassum got larger, it supported more individuals and more species than a smaller clump. But because we were looking at what we call those morphotypes of sargassum, so fluitans 3 and natans 1 and natans 8, we saw that even though that increase was present, fluitans 3 still supported more individuals per gram of sargassum than natans 8 did. So if our algal clump weighed 1 gram, fluitans 3 on average had more numbers of animals and more species than a 1 gram clump of natans 8. And 1,000 gram clump of fluitans is going to have more individuals and species than 1,000 gram clump of natans 8. So it's really important to sample both small and large clumps to make sure that that trend is the same across, the whole variety of sargassum that you could observe in the ocean.
Very cool. So definitely sample both to get a full picture. Yeah, that always makes sense. You said you found the different morphotypes of sargassum, and you just told us that they have different species associated with them, or like more and less species associated with them. Can you tell us a bit about the different morphotypes and how they make a different habitat?
Sure. So I guess this sort of brings me, can I tell a quick story? My colleague Jeff Shell, who works at Sea Education Association still, was sailing across the Atlantic. We, SEA, had not done transatlantic trips. We had typically sailed from Massachusetts to the Caribbean and back and forth that way in a north-south transect for many, many years, for decades. And he was sailing across the Atlantic from the Canary Islands to the Caribbean in November, December 2014. And if those of you that are familiar with the sort of new regime that's happening and the inundations that are happening in the Caribbean, this all sort of started to come about from sort of 2011 on, but 2014 was quite a big year. And he was sailing across and nobody was really sure what was going on yet. And he was standing at the rail, the way he tells it, standing at the rail with the captain and that he was sailing with, and they were looking at the sargassum, and they said, that doesn't look like the sargassum that we always see in the Sargasso Sea. It just floats differently. And so this is that years and years and years of experience of just observing sargassum and having looked at it and collected it and explored it with students. And so started looking, collected some, started looking more carefully at it, and started going back into the literature. And we referred to the works, early works, and one of the major ones that sort of we've used to move forward with is Parr from the 1930s, where he divided sargassum into pelagic sargassum into two species, Sargassum natans and Sargassum fluitans, but then kept and identified these different morphotypes within the species. And so, and indicated, had done lots of sampling and indicated which were most common in different areas. And we found that we were seeing on this transatlantic voyage, and then I sailed across in 2015 and saw the same thing, this dominated sargassum, completely dominated in the equatorial Atlantic by Sargassum natans 8, which looked really different from Sargassum natans 1, which we often see in the Sargasso Sea, and Sargassum fluitans 3, these are all Parr's morphotypes, Sargassum fluitans 3, which is frequently found mixed in the Sargasso Sea, but more frequently in the Caribbean and the Gulf of Mexico. And Parr said Sargassum natans 8 was not very common, but he found it. And all of a sudden, it was dominant all over the equatorial Atlantic. And those, in 2014, when Jeff was sampling, it was all Sargassum natans 8. When I sailed across in 2015, it was all Sargassum natans 8. And so how does it look different? Sargassum natans 8, I like to describe as sort of more rangy. It's has sort of long stipes, broad leaves, broad blades, but there's greater distance between the blades on the Sargassum natans 8 than on Fluitans 3 or natans 1. The big difference in telling natans from Fluitans is that Fluitans has thorns along its stipe, whereas natans is smooth along its stipe, and this is how Parr distinguished the two species. But then natans 1 is quite dense in its structure, similar to Fluitans 3, with long blades, and natans 1 has finer blades, but quite dense in its structure. And at the very beginning, when this natans 8 was washing ashore in the Caribbean, it was often confused for Fluitans 3 because of its big blades, which are more similar to the Fluitans 3. And we started to look at macroalgal references, and the most recent references had just sort of forgotten about the other morphotypes of Sargassum and just referred to Sargassum natans and Sargassum fluitans and described Sargassum fluitans 3 and Sargassum natans 1. So they had sort of forgotten to describe other forms that could be found. And so they were identifying this Sargassum natans 8. It was mistakenly identified as Fluitans 3. So what you can see, if you look at the three of them together, you can see this in our paper. We've got images of the three of them. And there's really just less interstitial space, less space between the blades for these motile organisms to find refuge and to cling on natans 8 versus the other two forms of Sargassum. And so we likely think that there's just this, that it's that difference in space between blades. It's the amount of branching that happens. There's much more branching on natans 1 and Fluitans 3. And actually, when we looked at some measurements with some students at SEA, we found that Fluitans 3 just seemed to have a much more complex habitat, even more so than natans 1, likely because the blades are broader and it has a greater branching structure to it. And so it really is the different types of space available in these three different types of Sargassum that may influence the communities that are found within them.
Yes, and just from seeing the different species from the beach, like in the water where you can swim in, I often find Fluitans as like a ball, really clumped up, while the others are a bit more broader for sure.
So that was nice. I really like the way you described that, and what you're saying makes sense about the spaces and interstitial spaces. When you start talking about interstitial, that things get pretty tiny. But if you got a lot of space in there, it can obviously hold more stuff and all. But tell me about the species that you most often found, and has there been a shift in that over time, or even seasonally?
So we had. It's interesting. We started diving back as we were looking at the communities that we were finding on these different morphotypes. We started diving back into the literature that we had been familiar with in sort of exploring Sargassum over the years. And a lot of the earlier studies looked at differences in communities between locations, or differences in maybe sizes of the Sargassum, or the age of the Sargassum. How does the community change? But nobody had really looked at the differences between the different forms of sargassum until now. And when we do look back on things, the community that from the 70s and up until the early 80s was much more varied. There were a lot of isopods. There were shrimp and snails, but it was quite varied. There were a lot of polychaetes and flatworms and amphipods, and it was a quite diverse community that was observed on the sargassum and recorded in the literature. But there seems to be a break somewhere in the early 80s, and there's a shift, and we see it in the reporting from a paper by Stoner and Greening, where all of a sudden the sargassum is dominated by the slender sargassum shrimp and the common sargassum snail, and really dominating the community. And we found the same thing, and other papers since the early 80s have seen that same thing, where these shrimp and the snails dominate over all other species that we find. For us in our study, the slender sargassum shrimp occurred on greater than 85% of the samples, and the snail occurred on 55 to 63% of the samples. And the shrimp was most commonly observed, but the snails were most abundant across our samples. Often we find tons of snails when we found them. And so
she says tons. We found a few samples that had like thousands of them, and we had to count all of them.
Yeah.
So there really has been this shift in the community, and we're not sure what has caused it. That's not what our study looked at. There are some ideas that it is related to potentially climate change, or maybe a change in predation on those communities, but we don't know. But there has been this shift in community.
Let me kind of follow up on that. Since Lindsay was talking about these related species exploit these mats for food and stuff. Did you also, in your lit review, did you also notice, or were you even looking at whether or not some of these apex predators that exploit these mats, did that change at the same time too? Was there a difference there, or was that even something you looked at?
So we didn't necessarily observe that. The largest organisms that we really counted were the small fish that were found kind of very close in sargassum. Part from a practicality standpoint, it's really hard to catch a large fish swimming under sargassum with a pool net. But also their movements, they're able to move much more freely than the things that we were looking at. So it's a completely different community. Like the mechanism that establishes the community that we looked at in this study is completely different than why, say, a mahi or a tuna is swimming underneath a sargassum clump. I know there have been several studies that have looked at associations of fish with sargassum clumps, especially in the Gulf of Mexico. There's just a higher concentration of fish underneath them there. But I don't think there's been too much research about if that change has translated to the higher trophic levels.
Thank you.
About these species, we have one more question coming from Mark. You found no true herbivores in the motul fauna, but are animals using sargassum as a refuge, or the sargassum itself? Is it being consumed? Are there any species consuming sargassum? Is the question.
Yeah. So to preface kind of where our trophic designations are coming from, there was a really excellent compilation of research from the 1980s around the Bermuda Biological Station. It's Butler. It's a book. It's absolutely excellent. It's got everything you needed to know about sargassum in the 80s. And he published in that book some observations of modal fauna consuming sargassum. So he observed things. He tried to do stomach content analysis. But when you're trying to do stomach content analysis on a thing that is like 2 millimeters long, not sure how accurate the results can be. So there isn't much research on the trophic ecology of the sargassum community. A lot of it, like I said, is based on these slightly anecdotal observations from Butler. There have been some stable isotope studies more recently, but again, they're really rare. But as far as we and Butler can tell, not much eats sargassum. It is fairly tough and unpalatable. A lot of algae are like that because they don't want to be predated on. Instead, the sargassum ecosystem, the sargassum community, is the primary consumers are detritivores, so things that are consuming decaying sargassum. So not necessarily the live stuff, but once it starts to break down, they'll eat it. Or they're eating the attached sessile fauna, so the bryozoans, the things that are crusted onto the sargassum itself. Or once you start to get to higher trophic levels, they're eating other modal fauna, or algae that grows on the sargassum itself, or even plankton from the surrounding water. But yeah, you are correct. For the most part, not much eats live, healthy sargassum. It's just not a good food source.
Very cool. Yes.
You kind of have to think of it as
like if
you're eating the thing that's keeping you alive. Not sure if that evolutionarily is a good thing. So they're not eating it. So it seems to work for them.
Yeah, I guess it's like eating your house. We don't really eat our houses either. And you can definitely see, like if you look at seagrass, for example, you often see bites out of it when you see it on the beach or you see it floating around. Or even if you go snorkeling and see the live seagrass. But with sargassum, it's always nicely intact. I don't see any bites taken out of the sargassum. So I guess there's really not that many herbivores that actually do eat it. My next question is about the changes we have seen, especially in the tropical Atlantic. You said there's a lot of sargassum natans 8 there. And that's not as, maybe not as ecological valuable as the sargassum fluitans 3 and natans 1, which were historically dominating sargassum mats. So from that, there's a risk that actually sargassum isn't that great of an ecosystem to live in anymore for the trophic resources and the food webs. But then we have so much more sargassum right now. So does that balance it out? Is it still positive or is it actually not that great?
Yeah, and that's the question that ecologists and economists and conservationists and everybody is asking. Well, not all of them, but a lot of them. Because, like you said, from an ecological point of view, you suddenly have this less ecologically valuable in terms of the diversity of species it supports and the number of species it supports, natans 8, morphotype that dominates everywhere. So is the negative effect of the fact that it's now a lot of natans 8, is that going to outweigh the fact that there's suddenly a lot of habitat? There's got to be some balance in there, but nobody knows. And, if there are specialists that live on just fluitans 3 or just natans 1, we did find a few species that were only found in certain regions or only found on a particular form of sargassum. But, for the most part, these species are able to move around. These clumps bump into each other. They're able to move between those clumps. It's not like we have completely different communities on natans 8 than we do on fluitans 3, but our study showed they're different enough to matter. So wherever that tipping point is of, suddenly we have a ton of habitat, that's great, more places for things to live, but it's not the best habitat. So where is that ecological balance point? And what's really important to consider is that if those changes do end up occurring, we've already seen this community shift, like Amy mentioned, since the 70s and 80s. If another one happens now, that's going to cascade. It's going to affect the higher trophic levels, the fish, the turtles, the seabirds that come to visit to sargassum. And that's going to affect, potentially important fish species that we humans rely on. It's not just, tiny organisms that rely on sargassum. And you kind of have the same dichotomous nature of sargassum for when it gets close to shore, both for people and animals that rely on it. When it's out in the middle of the ocean, I mentioned in the Gulf of Mexico, fishermen target sargassum because that's where all the fish are. When it's out offshore, sargassum is great. It concentrates fish. If you're a fisherman, you don't have to go trolling all over to catch a fish. You just go to a sargassum mat, drop a line in, and you will catch a fish much more quickly. But that same fisherman, as soon as that sargassum hits the shore, it is creating an anoxic environment as it decays. So, it's killing coral reefs, it's killing seagrass beds. It is washing up on shore and, potentially preventing beach access, both for tourists that provide income, or, you can't even get your boats out of some of these bays. So not only is there this weird balance of form versus, or morphotype versus quantity, but there's also this, sargassum offshore and sargassum onshore mean completely different things to the same person. And so from a conservation management standpoint, it's an incredibly complex issue, especially across the entire Caribbean, basically, and now starting to stretch up into the southern United States.
Well, and we're also seeing, I talked about 2014 and 2015 when Jeff and I sailed in consecutive years across the Atlantic and saw only sargassum natans 8. And now there seems to be a mixture of the three forms throughout the Caribbean region. And so even as we are working on this research and as we're studying the sargassum community, the makeup of the composition of the morphotypes and their distributions appears to be changing in that time. And so perhaps maybe this mixture of sargassum morphotypes is more beneficial than a single morphotype available, and particularly a single morphotype that may not be as ecologically valuable.
Yeah, that's true. When I was studying the sargassum on South Caicos and the Turks and Caicos Islands, we hardly found sargassum 8. That was the hardest one to find. And here in Mexico this year, we do have sargassum 8. We have all three species or all three morphotypes, but fluitans 3 is definitely the most common one I see. Like bigger clumps of, and the other ones are a little bit interspersed and also there.
For the study, you sampled in the Sargasso Sea, in the tropical Atlantic, all over the Caribbean and between the Caribbean and the Sargasso Sea and in the Gulf of Mexico. Did you see any difference between locations?
So we did start part of our analysis trying to also tease apart the regional differences in communities. With the nature of our sampling, different regions were sampled in different seasons, and also in different, I guess, spread out at different times across our sampling period. And so parsing down to the regional level really reduced the amount of samples in each region, and it was hard to sort of tease apart differences. But one thing that did stand out in our analysis was, first of all, the Sargasso Sea. Most frequently, that's where we found Sargassum natans 1. And Sargassum natans 1 has its sort of specific rare organisms that seem to be specialists on Sargassum natans 1. Like one of the crabs, the pycnogonids seem to be more common there, and things like that. Then also, what also stood out was the Gulf of Mexico and the association of fishes with the Sargassum in the Gulf of Mexico. And we posit that that's likely due to being sort of more close to shelf region and coastal environment, maybe where larval fishes and other fishes may come and aggregate toward the Sargassum more easily than a lot of our offshore sampling locations, or farther offshore sampling locations. So we did see some differences, but harder to tease apart in this data set.
Well, nice. Thank you. One reason that you were able to collect these samples in all these different locations is because y'all were, for lack of a better word, running around the Atlantic playing on the sailboat Corus Kramer operated by the Sea Education Association, which got to be, we really appreciate you suffering yourselves through this to do this important research. And that's a novel way of doing your research, and especially when you take into account that you're also, it's a teaching vessel, and you're working with students. Can you share a little bit about your travails as you were floating around the sea, teaching students and making all these collections?
Sure. Yeah. So, like I said, SCA was a formative experience for me because it was, I for several years up till that point, been working in, biology. I was actually studying ferns in undergrad, which was a great, field experience and learning how to conduct science. But I thought I wanted to go into marine science. And SCA was my first chance to test that. This is not your typical research vessel where you get on, there's a ship crew that does everything for you. There's a chef, there's a cooking team that cooks everything for you, and you just do the research at certain times of the day. With SCA, you are part of the crew. You are doing sailing operations and navigation. You are helping to cook. You are cleaning. You are doing research 24 hours a day. It is a truly amazing experience. It's really, really hard. And all the better for it. But I was out there sweating. There's no air conditioning, sweating and covered in like sargassum that we were sorting and slightly seasick and sitting there and looking up and realizing I absolutely love this. This is great. So for me, sailing with SCA was confirmation that this is in fact something that I really love and want to dedicate my life to. And the cruise that I went on with SCA was in 2012. And unfortunately, the samples that I collected in the Gulf of Mexico were not done with SCA. That's kind of why I was brought on to this project, because we wanted to sample that area, but SCA doesn't go to the Gulf of Mexico. So I collected my samples by reaching out to professors and research groups in the area who had cruises scheduled in the Gulf of Mexico, and just grabbing the last remaining spot on their cruise. And for me, that was also an interesting experience, because I got to go on cruises that were completely not dedicated to sargassum. One was sampling deep water in the Gulf of Mexico in response to the Deepwater Horizon oil spill. Another was doing ROV surveys of corals, and another one was looking for sargassum hatchlings in sargassum. So it was not only an opportunity for me to, do this research and share what I was doing with people who had no clue about sargassum, but also to participate in these other research projects. So for me, this was another very cool experience.
Awesome.
Very cool.
Those are, I think those are the two best answers we got out of everything today. Thank you for sharing with us. Well, that concludes our interview for the day with Lindsay and Amy. This has been, just like everything else, this has been a very interesting conversation that we've had, and I really appreciated this because we got some of the nuts and bolts of what sargassum actually is, instead of some of these remedies and all this stuff. So this has just been a wonderful, wonderful talk today. So Lindsay and Amy, thank you so much for being with us today, and we hope to talk to you again real soon.
Thank you so much for having us. We really appreciate this chance to share our research.
Hello. So what did you think about this beautiful interview about the Matal fauna?
Well, a lot of our interviews are about technical things and things that people are doing on shore to alleviate these things. But much like our interview with the Sargasso Sea Commission, this was about sargassum in its native habitat and in this natural world. And I just loved it. It was wonderful to discussing the importance and the fundamental ecosystems themselves, as opposed to some of the other stuff we've been talking about. It's been a pretty happy interview, I think.
Yes, I agree. It was
Exactly.
so nice to hear about the positives of sargassum and how amazing it is out there, and that having more of it is maybe a good thing, as long as it doesn't get to the beaches. So I think for us who are suffering from the sargassum on the beaches, we have to find a way to collect it, but not too far offshore. So we keep this positive aspect of sargassum there, and just make sure it doesn't negatively affect the beaches.
Yeah, I totally agree. And they start the interview with this, that it is impressive to have Matal fauna in the middle of the sea, in the middle of the ocean. So when we think that it is thanks to sargassum that we have those species in the middle of the blue, it's like, wow. And actually that it is their home and they don't eat it. But yeah, it's beautiful. And I also laugh a lot when they explain how they sample it and what they do after, how they process it. They just put the sargassum in the fresh water and all the species go out. It's like, oh, it's not cool. And I find it funny. Anyway, it's like traveling when we hear the story about their expeditions.
Yeah, like those were really good questions from our Patreon Mark, who actually brought out these things that they talked about. And when they told us that they would find thousands of these, I think it was shrimp, and that they had to count all of them, like I can imagine those students on the boat, they're just spending hours and hours looking at sargassum. So that paper has so much work behind it and so much depth and also so much knowledge. Like Amy told us that she has traveled those waters years after years, the same place. So she is pretty much a local of these waters as much as you can be, like other people are from the coast who live there and observe every year what's going on. So I think that's really, really valuable.
Yeah, and she can notice the changes of species and morphotype and abundance and diversity and everything, which is really precious.
Yes.
Yes, it was. And everybody, we want you to know that we appreciate you being here with us today. You could have been anywhere on the planet, but you chose to be here with us, and we appreciate it. And until next week, this is Robbie, Cleo, and Franziska, and we'll see you next time. Thank you much.
Thank you for tuning in today and learning with us from our guests. If you want more information about what our guests talk about today, then please check our show notes for links and information in our archives. And don't forget to like and share our podcast with your friends.
If you enjoyed our podcast, please consider supporting us financially by becoming a Patreon. For as little as $1 per month, you can support us and get the exclusive benefit of submitting questions for our interviewees before the interview.
The Sargassum Podcast is produced by Marine Conservation Without Borders and is made possible with financial support from Seafields. It is produced by Marcel Van de Kamp, Lauren Blankenship, Cleo Maridakas, Franziska Elmer, and Eloise Lopez, and is hosted by Robbie Thigpen, Franziska Elmer, Mar Fernandez, Florence Menez, Cleo Maridakas, and Paula Diaz.
We will be back next week with another exciting guest. The music of this podcast is from the song Demapre by Drizzle the Roadrunner, an artist from Roatan. Follow him on Spotify or YouTube for more music, but for now, here is the full song, Demapre.
Hey, brother, hear me now. Brother, dog. Now me understand. Now for them no want to see we get nothing. That's why them a pray and always front and star. Now for them no want to see we get nothing. That's why them a pray. Now for them a pray. Them a pray. Them a pray. Me no gain progress. Now for them a pray. Yeah, man. Them a pray. Me no great success. Now for them a pray. Them a pray. Them a pray. Me no gain progress. Now for them a pray. Them a pray me to reap success. So me tell them, yeah, promises come and me no take that. Only if it come from Jah, I'll accept that. Not for them me put me trust in and give me setback. Yo, select that, will and pull up that. Tell some wicked that bad mind me no fear them. Anytime them cheat and chat, me no hear them. Me dash a few hearts so for the queer them. Me dash a few hearts so tell them where them. Not for them a pray. Them a pray me no gain progress. Not for them a pray. Them a pray me to reap success. So me tell them, yeah. Yes, me know me have a lot of fake friends, but me never woulda thought me woulda have fake family. So me tell them straight, me no trust them. Me no trust you and me no trust him. Fake friend lost bad mind in a real life. Star me no rate that, star me no rate that. When me real for me woulda bust a million shot in a real life. Now for them a pray. Them a pray me no gain progress. Now for them a pray. Them a pray me to reap success. Now for them a pray. Them a pray me no gain progress. Now for them a pray. Them a pray me to reap success. So me tell them, yeah. Oh, them a pray me to reap success. Screaming out loud. Me woulda tell them to go look alike, but them a hate and grudge and creep on mine. Them a move like Judas. Them a move like Judas. Plus, everybody have a life to live. So when I give one rascal who a try judge me, like them chit and chat. So what them want to say, cause none of them out there nah feed. Now for them a pray. Them a pray me no gain progress. Now for them a pray. Them a pray me to reap success. Now for them a breathe. Them a breathe, me no gain progress. Now for them a breathe. Them a breathe, me no reap success. Oh, five-star production. 1101. Them a breathe, me no reap success.